My topic today is about sense and adaptability of animal cells to oxygen availability. Now, the general buzz this year around the 2019 Nobel Prize in Physiology and Medicine had been, which one awarded to William Kaelin, who is attached to the Harvard University and Dana-Farber Cancer Institute, Peter Ratcliffe, who is attached to the Francis Crick Institute, and Greg Semenza, who is attached to the Johns Hopkins University. So they were basically given this Nobel Prize because they cracked the code how our cells sense oxygen availability in and around them. So just going back a little with the discovery of oxygen, I'd like to take you through that Sheely and Priestley almost simultaneously discovered oxygen. But because Priestley published earlier, which is again a lesson for all of us scientists that we should publish as fast as we make a discovery, he is credited to being the discoverer of the oxygen. And if you do remember, a number of us in our biological textbook did study about Priestley's bell jar experiments, where he put in a mouse and a burning candle and the mouse died. Then he put in a mouse and a burning candle in a plant and the mouse survived. But he did not exactly coin the term oxygen. It was later on Antoine Lavoisier who coined the term oxygen. Now, when did oxygen become fundamental? As we all know, that oxygen is required for production of energy in our body. But oxygen is also required in certain of these genes that are regulated in the body. Oxygen is also required for that. But the atmosphere way back was not oxygenated. So what happened was 2.5 billion years ago, cyanobacteria decided to make their own food by photosynthesis. And in doing so, they started releasing oxygen as a byproduct into the atmosphere. And evolutionary biologists mentioned this place as the great oxidation event that occurred between 2.5 billion to 2 billion years ago. The moment there was oxygen in the atmosphere, what happened was there was almost 2 body-sized jumps according to evolutionary biologists, and there was a blasted evolution happening. So, and from single cell organisms, multicellular organisms with more complexities started evolving throughout the geological time scale. So now that we've established how oxygen became a part of our life, the next thing I'd like to describe is certain physiological conditions that may be pertaining to oxygen availability around us. Currently, all of us are sitting. We don't feel any uncomfortableness. We are having enough oxygen which is reaching to every part of the tissue of our body. This condition is normoxia. Let's say this hall becomes too crowded and there is not proper ventilation in this hall, and we are competing to use the oxygen molecules present in this hall, and after some time a lot of us will start feeling uncomfortable, dizzy, and there will be not enough oxygen reaching to every tissues of other cells. This condition of low oxygen level is hypoxia. There might be total sucking out of the air from this hall, and that might be anoxia, which no oxygen molecules. There is a condition called hyperoxia. Excess of oxygen may be present in and around you. This is something I'll touch up later in my talk. Another condition may be physioxia. Say there's an anemic person, doesn't make enough red blood cells. Even if there is enough oxygen around that person, he's not being able to utilize that oxygen, and hence some of his tissues may be suffering from hypoxia or low oxygen level. So low oxygen is definitely a concern. When can we experience low oxygen? If you're doing rigorous exercise, some of our tissues will be facing low oxygen problem. Mountaineers, trekkers that go to the mountains, even there were news recently that 11 people died while waiting in the balcony to climb the Everest because they had been spending too much of time at the dead zone, which is around this sector, for more than 24 hours, and that is a death nail to the coffin because they all died of exhaustion due to low level of oxygen. And in recent cases, hypoxia has become a marker for a very aggressive cancer prognosis. So with all these data, there was something that in the 1990s people started to focus on. It was erythropoietin gene. Now this encodes for a hormone, which in cases of low oxygen level in the body or say hypoxia-like condition would actually stimulate red blood cells to multiply in the bone marrow. So when Greg Semenza wanted to take a look at how this oxygen sensing is happening within the cells, his first focus was the EPO gene. And what he found out that there is a region in the EPO gene which probably is oxygen sensing. Peter Ratcliffe was also working on the EPO gene. Now Greg Simmons are working on, since EPO was first found in kidney, he was much more interested in the kidney cells. Peter Ratcliffe was actually working on transfected hematoma cell lines. But what both of these scientists found out eventually, that this is the universal scheme in every tissue, every cell of the body. So the oxygen sensing happens everywhere in the body. So Greg Simmons took a much more deeper look into this gene. What he found out was that in the 3' flank of this gene, there is an enhancer region of 50 base pair, which was basically detecting the various nuclear factors, transcription factors bounding to it. And he named this gene as the hypoxia responsive element. Among the factors binding to this place, he found out that one factor was basically oxygen dependent. So if the cells were having enough oxygen, it was not binding. And if the cells were having lack of oxygen, it was binding. And he purified this factor. He called this hypoxia inducible factor. And he found out that this is a heteromeric complex having one alpha B and one beta. And he found out that 1 beta, HIF1 beta, is basically a constitutive factor. So it's present in the cell. It is the 1 alpha that is actually oxygen dependent. In fact, post-translationally, it undergoes oxygen dependent degradation. So what next in this puzzle? The next thing was to find out who exactly was doing this oxygen dependent degradation to the HIF1 alpha. And how was it commencing? Like any good thriller novel, there are parallel storylines that develop with the story. And William Kaelin at this time was looking into von Hippel-Lindau cancer diseases. And while doing so, he found out that this is VHL, I'll just use this short form, is a tumor suppressor gene. Following on his work, other groups found out that VHL actually is a part of a bigger group of proteins which have this tendency of tagging protein with ubiquitin, which would later go on to proteasomal degradation. So could this be the factor that we are looking for, which were binding to the HIF1 alpha and degrading it? Ratcliffe made a milestone discovery at this time, that HIF1 alpha and VHL were indeed physically interacting with each other. Now let's see how the missing pieces of the puzzles fall. So here we have the VHL binding to HIF1 alpha, ubiquitination happening and taking this towards proteasomal degradation. So how did VHL bind to this HIF1 alpha? Kaelin and Ratcliffe together discovered that there was an enzyme called prolyl-4 hydroxylase. That was basically in presence of oxygen, induce hydroxylation in proline residues in the HIF1 alpha. Now Simmons had found out that HIF1 alpha has an oxygen dependent degradation domain, and this was in this domain this proline residue was getting hydroxylated, and Kaelin and Ratcliffe discovered that hydroxylated HIF1 alpha has a more affinity to bind to VHL. So finally we have the scheme. In presence of oxygen, enough oxygen, that is a normoxia condition, the prolyl-4 hydroxylase will actually will induce hydroxylation in HIF1 alpha. The VHL will recognize this, bind to it, and will take it to proteasomal degradation. When there is less oxygen in the cell, like hypoxia condition, HIF1 alpha goes unhindered to the nucleus, binds to HIF1 beta, and would continue transcription of hypoxia regulated gene. Now this was the scheme of when there is low oxygen. I also spoke about if there is a hyperoxia case. We do not usually face it, but remember 2.5 billion years ago, the Earth's atmosphere did not have oxygen. And there were animals who were anaerobic animals and they were surviving pretty well till the cyanobacteria decided to do a different take. And at that point, the oxygen started accumulating. Now this was not a one-day event, it accumulated almost 0.5 billion years. And at each stage, there were animals who either adapting to this increase in oxygen concentration or were actually dying because of this. During the Great Oxidation event, Earth actually saw its first mass extinction. Millions of organisms died who could not adapt to this changing in oxygen concentration. If you take a look at the table, you would see that as atmospheric oxygen started rising and reached our current composition, there was again a burst of evolution almost from dinosaurs to humans. And as we were going up, organisms were evolving, complexities were increasing, but the evolution was a little slow. So oxygen is definitely fundamental to all of us, but it also has bad effects. Oxygen derivatives are not really good for any one of us. So how was the animals who were surviving this accumulation of oxygen in the atmosphere able to imbibe that oxygen into them and evolve and proliferate? So here comes a very interesting concept. A number of evolutionary biologists do believe that sterols acted as oxygen sensors at this point. So the question was sterile induction a feedback loop curated by natural selection to protect organisms from the biotoxicity of oxidative derivatives? A look at this picture would tell you that how the sterols have evolved with the accumulation of atmospheric oxygen. So maybe this could be a reason how these organisms coped up, and also this threw off much more complexities in the network, and which may also be a reason why more complex animals started developing after the Great Oxidation event. In fact, Conrad Bloch, who was awarded a Nobel Prize for elucidating the cholesterol pathway in 1964, mentioned famously that he thinks that cholesterol is a superior sterol. And if we do think that vertebrates are evolutionary success, then yes, cholesterol is there. And a look into the cholesterol pathway will actually tell you that it is a very oxygen sensitive pathway. From larostrone to cholesterol, a number of molecules, 9 oxygen molecules are used, and if there is hypoxia, larostrol accumulates and takes to degradation of the HMG-CoA reductase in its synthesis as a feedback. So coming to the conclusion, we see oxygen regulating genes. Oxygen being a part of the cholesterol biosynthetic pathway, it helps in generation of blood vessels. Cells adapt metabolism to changing oxygen levels, and it also fine-tunes immune system. So coming to the conclusion, this helps us in devising, this mechanism would help us in devising and having therapies against diseases that are a product of low oxygen levels, for mountaineers, for even soldiers posted in Siachen who take in effects of low oxygen, therapy can be developed for them. And it's very exciting, this particular work is very exciting because I as a postdoc for the past 1 year had been working on how hypoxia affects the brain, and specifically the serotonin 1 receptor, which is one of the major neuroreceptors in the brain. Thank you all. I'll be ready to take in questions.